Am I audible and my slides are visible?
Yes, ma'am.
Okay.
Yes, ma'am.
I have started the recording. Okay. So I was just analyzing the syllabus of whatever we are having. So in the third module, so we have total of 5 modules. So I analyzed it in a very detailed way. So I found that module 3 is one of the most important module, because module 1 was basic, related to what is microbiome, what is the composition of different bacterial and fungal group, and what is the actual role. Like, it was an overview, basic thing. And in the second module we have gone in a bit more detail, but in the third module we have read in so much detail that how the gut microbiota is associated with different gastrointestinal disorders and disease. And specifically we have read in detail IBS, IBD, and celiac disease. And we have also studied diabetes, obesity, and this liver disease. Now we will also study autoimmune and neurological disorders, like how the gut microbiome or gut microbiota is associated with different autoimmune and neurological disorders. And apart from that, the major one is cancer. So this unit is one of the most important unit of out of all the modules we have, because after that in module 4 we will be studying about the therapeutic approaches, like if our microbiome composition is disturbed by any of the factors, so how we can revert it, how we can opt any of the available therapeutic mode, so that we can compensate whatever we are not having naturally in our body. So we have to understand this module 3 very in very detail, then only we will be able to uh, grasp the therapeutic approach. And if you are able to grasp each and every topic of module 3, then you will be able to uh, state that what is the actual role of the gut microbiome in controlling our normal physiology or normal metabolic machinery going on in our body. So we will study it in a bit detail. So we have already covered the first part, and the second part we are in this one, NAFLD. IBS, IBD. We have to cover from here. So gut microbiota as a predictor of diagnosis or biomarker of any metabolic disease, including diabetes. So why they are acting as a biomarker? Because if we are disturbing our normal natural composition, then there may be increase in the bad bacteria, and then that bad bacteria will act as a marker, and it will denote that there is something going on in our body. And it will also denote that our body is having altered microbiome, because the concentration or the population of the good bacteria had declined and the bad bacteria is already increased. So this diagram shows that how our body is associated with insulin resistance and how our body is involved in different inflammatory responses. So we have to just reduce the inflammation in our body in order to prevent different kind of metabolic diseases. So dysbiosis and diabetes pathogenesis, increase in gram negative species and high fat diet led to the translocation of bacterial lipopolysaccharide. So this LPS is acting as a inflammatory mediator. Inflammatory mediator, because LPS is secreted by gram negative bacteria as an endotoxin. So this endotoxin is responsible for generating a inflammatory response in our body, because whenever our blood will encounter any endotoxin secreted by any of the gram negative bacteria, then it will consider it as a non-self. Obviously they are non-self, so they will start activating their own immunological uh, pathways like complement pathways to kill or neutralize the effect of endotoxin, because this endotoxin has a potential that it will bind to your any of the biomolecule or macromolecule, and then it will start the signaling cascade and it will have some bad consequences in your body. So your immunity, if your immunity is intact, what it will try, it will try to just bring down the concentration of LPS by neutralizing it. It will activate the macrophages, it will activate the antibody production, it will also activate the complement pathway. So through so many different ways, your immunity will try its best to remove or eradicate this LPS, because this LPS is acting as a inflammatory mediator. Now, so what are the major factors you can say which are responsible for diabetes pathogenesis? I have yesterday also discussed about the it is a metabolic disease. Metabolic disease means you are only the responsible for the diabetes pathogenesis, because the major role is of diet. And nowadays our diet is not at all clean. It is full of junk and overload of only glucose, only sugar. We are taking only sugary diets. Whenever we are feeling hungry, we are always in a search of any junk food, any packed item, any processed food, which has a high amount of sodium and a very high amount of sugar in it. And apart from that, if our body has a certain kind of infection, so it will have increased amount of gram negative bacteria. So both these factor, like the increase in the population of gram negative bacteria, as well as if we are consuming a diet which is rich in high amount of fat and carbohydrate, so it will ultimately do what? It will increase the lipopolysaccharide in your blood. It will directly bath into your blood, and then what it will do, as you all know, if our metabolic machinery is hindered, if gut dysbiosis will occur, what it will do? It will inhibit the production of short chain fatty acid, specifically butyrate. As simple as that. This key point you have to mention it in your copy. Like I used to mention it whenever I used to make a notes. So suppose this is your notebook. These are the two pages, right and left. So what are the whatever be the key point, I used to write it on a top of the page, so that I can, whenever I will open the copy, I will just focus on these points, and rest of the details of that topic I used to write it in the bottom. So whatever be the crisp note, don't write a kahani or a story. Always try to make a crisp notes, bullet points. Like what I told, that whenever there is an increase in the LPS, there will be inhibition of the short chain fatty acid production. So write it like this way. If LPS will increase, it will inhibit SCFA. No need to write long sentences, a paragraph. This is very simple and it will take only few seconds to memorize. And if you are writing it, you will remember it. This picture will be memorized for a longer period of time. So try to write all the crisp points on the top of your notebook. Now back to this topic. So if LPS will increase, it will not allow the, because already you are having a bad amount of bad bacteria in your gut. So they will not allow your metabolic machinery to synthesize butyrate, that is the short chain fatty acid, and ultimately you will be facing insulin resistance. No insulin will work in your body. So insulin do what? Insulin converts the excess of glucose into glycogen, so that it can be used whenever your body is in fasting mode. But this phenomenon is hindered whenever your body is having low production of insulin, that is type 1 diabetes. And if you are having insulin in a sufficient amount, but your cells are not able to use that produced insulin, that condition is known as insulin resistance. Your cell are not capable of sensing that produced insulin. So how they will convert the excess of glucose into glycogen? So this is the reason insulin resistance or the type 2 diabetes mellitus is nowadays increasing, and every 3 out of 5 people are suffering from type 2 diabetes mellitus. And nowadays it is very common in adults also. So these are the responsible agent for increasing the inflammatory mediators in our blood. So the main consequences of diabetes associated dysbiosis that affect gut wall integrity, leading to endotoxemia and chronic inflammation. Whenever our body will face chronic inflammation, your all energy will be drained. Whenever we are sick, so you are always feeling lethargic. You don't have that energy to do any of the tasks, like you just want to sleep and rest. Why this is so? Because all your energy is drained in providing the inflammatory responses. So all your ATP is involved in the inflammatory responses. So we have to conserve that ATP so that we are having that much amount of energy to do our normal day-to-day life activities. So we have to, in order to remain healthy, fit and active, we need to bring down the inflammation of our body. Like many of the herbal therapeutics targets to bring down your inflammation in your body. First of all, they just analyze that whether your body is inflamed, whether some uh, different reactive oxygen species are involved in initiating the inflammatory responses in your body. So if there is an inflammation, firstly they will treat your inflammation level, and then they will treat your another diseases, because no such treatment will work fine if you are having a high inflammatory responses in your body, because your body or the cells are already busy in something else. So how they will respond to your therapeutic approaches? So reduction in butyrate level also leads to insulin resistance because of reduction in glucagon like peptide pathway activation in fatty acid oxidation. So I have already mentioned one metabolic pathway, that whenever butyrate is hindered, suppose it is very low or it is not at all produced, so what it will do, the fatty acid oxidation will also be inhibited in the mitochondria. So this is written here. So fatty acid oxidation will be decreased. And what will be the consequences of it? If you remember, if the fatty acid oxidation is reduced, then increase in the oxygen concentration in that particular area. In your gut, oxygen concentration will be increased. Now, if the oxygen concentration will increase, are the obligate anaerobes able to grow? No, because they want strictly anaerobic condition. Not even a traces of oxygen should be there. So it will start facilitating the growth of the facultative anaerobe, which are the bad bacteria in our gut. So all these things are interlinked. If you will understand the basic, then you will be able to link it with any of the prospects. Like if you want to relate this basic thing, this metabolic pathway, to gastrointestinal disease, then also it will go fine. You can relate this to obesity and diabetes. You can relate this to even neurological disorders, because this is the only metabolic thing going on in our body. Whenever we are not producing the short chain fatty acid, our level of fatty acid oxidation will be low, and increase in the oxygen concentration in our gut. So what it will do, it will increase the bad bacteria. And what bad bacteria will do, the bad bacteria will synthesize lipopolysaccharide or other toxins. So when they will synthesize the toxin, our body will again start inflammatory responses. So everything is related to each other. So I hope this is clear. Now next part. So you can see non-alcoholic fatty liver disease. So this is a very common, and again adults are very prone to this fatty liver disease, because they are involved in eating so much of junk. Junk is the only reason for this NAFLD. The most what you can say crucial factor is eating junk and not doing any kind of physical activities, physical exercise and all. And then we are having a sedentary lifestyle. So all these things are related to each other. In our research, we used to say that like we are lucky that we don't have any sedentary job or anything, because we have to work a lot in research laboratory. So we don't have a like a 9 to 5 job. We have to work in the yes, we have to work, we have to write paper, but that is only a uh, shorter duration of our day to day life activity. But most of the time we are involved in doing experiments. We are visiting one lab, then second lab, third lab, like this. So whenever we are eating junk, whenever we are not doing so much of activity, whatever we are consuming, we are not burning it. So then there will be a accumulation of high amount of fat in our body. Then it will again start inflammatory responses. So NAFLD is characterized by excessive intrahepatic fat accumulation. So whenever there is an NAFLD, your so many profiles will be altered. Like your total cholesterol will be altered, your bad cholesterol will increase, and your good cholesterol, that is HDL, will decrease. So it will ultimately result in fat accumulation. You can see in this diagram. So why this is known as non-alcoholic fatty liver disease, I have discussed this previously also, that this is not related to the significant alcohol consumption. Like you are having this non-alcoholic fatty liver disease. Why this is non-alcoholic? Because no, this is not dependent on the alcohol consumption. If a person is not consuming alcohol in a significant amount, or he or she is not at all consuming, then also he or she can have this disease NAFLD, because this depends on the fat accumulation, not at all dependent on the alcohol. So that is why it is to characterize between the alcoholic liver cirrhosis and the normal liver disease. So the term is added to this normal fatty liver disease, non-alcoholic fatty liver disease. This is more common than the liver cirrhosis, because it occurs when you are consuming alcohol in a very high amount. So that is not the case with adults. So liver disease is defined as fat accumulation in more than 5% of hepatocytes. Hepatocytes are what? These are the liver cells. So it can be non-alcoholic, and if a person is taking alcohol, then again these consequences will be more fatal than the non-alcoholic one, because alcohol will also increase the fat accumulation in your liver. So see, this is the intestine, and you are, if you are having dysbiosis, it means you have altered the natural condition, altered natural microbiota. You have destroyed the natural microbiota. Then what it will do? It is termed as dysbiosis. Now dysbiosis will do what? Altered metabolites metabolism. Like it will not produce the required amount of metabolites, and it will not produce the required amount of good short chain fatty acid, and also it will synthesize some biomolecule which will act as a inflammatory mediator. So you need to understand the overall pathogenesis of the NAFLD. So you can uh, just read this point, that whenever there is a dysbiosis, these gut microorganism will have bad bacteria increase, and good bacteria will be decreased. Now if the bad bacteria is increased, it will inhibit the this one short chain fatty acid production, and then what it will do? It will start inflammation, and inflammation will do what? The accumulation of fat cells, fat accumulation of fat, and then this condition will be worsened day by day if you are not seeking any medical help. So this results in different hepatocellular carcinoma, liver cancer, liver fibroids, cirrhosis. All these things are related to different kind of inflammatory responses that is generated by the bad bacteria whenever you are facing dysbiosis. So what are these are the therapeutic approaches. These are the therapeutic approaches. You can have prebiotic, you can have probiotic and symbiotic and FMT, fecal transplant, fecal matter transplant. So you can have any of the option. We will not discuss this in detail in this module because we have to individually focus in the module 4 on what are the treatment options available if we are having dysbiosis. So we will not waste our time in this one. We will understand the pathogenesis of different disease related to the altered microbiota or dysbiosis. So NAFD is also closely associated with many features of metabolic syndrome like obesity, insulin resistance, hyperlipidemia, like increase in cholesterol or lipid is known as hyperlipidemia, hypertension, increased risk of CVD, cardiovascular disease, and type 2 diabetes mellitus. Now this NAFLD, if you are having, like whenever you are complaining about the liver issues or liver disease, your doctor will always check your all these parameters. Like he or she will not just check your liver profile test. No, he will ask you to do the this cholesterol level, cholesterol profile, lipid profile test, and your BP will also be checked. Your CVD markers will be checked, and also your blood sugar level, glycosylated hemoglobin will be checked. So all these parameters are interlinked. You cannot say that I have only liver disease because all these secondary complications are associated with type 2 diabetes. Likewise, if you are having a diabetes positive, if you are having high blood glucose, and you cannot say I only have a sugar disease, I only have a sugar. You can have the kidney nephropathy, you can have blood vision, you can have arthritis, you can have so many other diseases as well, hyperlipidemia, and also you can have the liver uh, malfunctioning. So any of the disease is possible. So nowadays you cannot say that I am only having this disease, I am only having that disease if you are suffering from any kind of metabolic disease, because all these diseases are interlinked to each other, because each organ is associated with the functioning of another organ. Your liver, your kidney, your heart, every organ functions in a very coordinated way. So you cannot say that my only this part is influenced, my only this part is diseased one. No, everything is interconnected. That is known as physiology. Your excretory system, your digestive system. If you are saying that my digestion is good but my excretory system or urine formation or the kidney is not functioning properly, so you are wrong, because everything is dependent on your what you are consuming. So if you are not taking clean diet, how your kidneys will be able to filter out the waste material from your blood? So you cannot say that I have digestive disorder, I have kidney disorder. No, both these things are interlinked. So now if digestive and excretory system you are saying about, so you cannot forget the cardiac, because if your digestion is not proper, your excretion is not proper, so your blood will be having only waste material. And if it is full of waste material, it is, if it is full of ammonia and it is not secreted out from the body, so then it will also influence the cardiac output, your cardiac system, your heart, your veins, your arteries, everything will be influenced. So this is known as the physiological system. Your body is having different kind of physiology, and if your digestive system, if your excretory system, if your cardiac system is damaged or influenced, then how can you forget about the nervous system? Because this is the brain. How you can think of that your brain will function properly if your stomach is upset, if you are not having good kidney function, and if you are not having a circulatory system, proper running circulatory system, then how can you expect that your brain will function properly? So what I'm trying to say that everything is interconnected with each other, and if your gut microbiome is altered, that is known as dysbiosis. I always used to the definition of dysbiosis and the term dysbiosis so that you can memorize it. So dysbiosis is nothing but the altered microbiota, all the or you can say perturbed or disturbed. If you are disturbing the normal gut microflora of your body, then it is termed as dysbiosis. So if dysbiosis will occur, you are ultimately targeting your digestive system, because gastrointestinal, obviously digestive system will be influenced, and if digestive system is influenced, then you can say your excretory system, which is the main physiology of the body to expel all the toxic substances or the harmful waste material from our body, because if they are in our blood, they may cause different kind of ailment. So, and if this our blood is impure, then it will lead to different cardiac diseases, and if all these three are affected, our nervous system will also be affected. So everything is related to each other. Gut microbiota alteration associated with NAFLD are dependent on the clinical stages of the disease. The most typical characteristics of disease progression include the decreasing microbiota diversity. So we have studied about the dysbiosis level 1, dysbiosis level 2, dysbiosis level 3. So when dysbiosis begins, what it will do, it will have altered microorganism composition, like it will start decreasing the diversity. In a healthy gut, A, B, C, D bacteria were there. Now it will only have A increased, B increased, and no C and no D. It means the diversity is decreased. So what it will do, it will increase the population of a specific type of bacteria, and that will always be a bad bacteria, negative bacteria. Negative means function wise negative. So which are the villain? Gram negative bacteria will increase, mostly proteobacteria, and a decreased abundance of gram positive bacteria, mostly firmicutes. So we have read that F by B ratio, firmicutes by bacteroidetes. So if firmicutes are going down in our gut microbiome, then something is happening in our body. Some inflammation is going on inside our body. So how these things are measured? So your doctor will conduct stool test. If our stool is rich in methanogens, then also our body is having inflammation. And if the firmicutes composition is also low, population of firmicutes is also low, then you are also having a trouble in your body. So how the things start? The things start with the decreasing diversity of the gut, and specifically gram negative bacteria increase and gram positive bacteria, specifically firmicutes, decrease. So there is also a shift from beneficial to harmful microbes. What I used to say, bad bacteria will increase and good bacteria will decrease. So this is the thing going on, leading to the development of a pro-inflammatory. Then it will do what? It will initiate inflammatory responses. So this key point you can write whenever there is an increase in the bad bacterial population. What they will do, they will release the inflammatory mediators, and this is enough to initiate the overall inflammatory responses by our own immune system. So they also secrete, apart from the pro-inflammatory molecules, they also secrete different toxic intestinal environment endotoxins, which will result in gut barrier dysfunction. Gut barrier dysfunction means when this cell is connected to this cell through the tight junction, then this tight junction will be not functioning properly, then this is known as leaky gut. So this is known as gut barrier function. So the exposure of liver to dietary and microbiota derived factors and NFLD progression. So all it depends on the dietary intake and how that dietary intake is disturbing your natural microorganism present in your gut. So if you are altering their composition, if you are decreasing the composition of bad bacteria, and increasing the composition of good bacteria, then you can have NAFLD, because all these things are related to diet, and bad bacteria because they are initiating the inflammatory responses. So this was about the gut microbiota is associated with NAFLD. Now some more detail. The most consistent gut microbiota signature associated with NAFLD are increased proteobacteria, Enterobacteriaceae, E. coli, bacteroids, Doria, and apart from that, you can see this one. This we have already summarized in a table. Rhininococcaceae, Faecalibacterium, Coprococcus, so Anaerosporobacter and Eubacterium. So you can write it down, all these names of the phylum, family, because all these species, you can say not only just species but different genus increase in the condition of NAFLD. This has been proven through different research, through the reports of different patients suffering from NAFLD. So these bacteria will be increased in the patient having the liver disease, non-alcoholic fatty liver disease. So this NAFLD associated microbiota signatures partially overlap with other metabolic diseases. For example, the level of Faecalibacterium are reduced in cirrhosis, as well as obesity, type 2 diabetes mellitus, or intestinal disorders such as IBD or IBS. And Faecalibacterium is considered a beneficial microbe for its anti-inflammatory properties. So now this is the marker. I was talking about the marker. So a normal pen marker do what? It is used to mark something, label something. So likewise, bacteria can also act as a marker. If any specific type of bacteria is increased, then you can it a condition. If it is decreased, then also it has some condition. So in NAFLD associated microbiota, the and also with the other diseases, you can expect the reduced level of Faecalibacterium, because this Faecalibacterium is associated with what? This particular strain is considered a beneficial microbe for its anti-inflammatory property. So you can expect a decrease in the good amount of bacteria. So bacteroides vulgatus, which is increased in advanced fibrosis, is associated with severe obesity, insulin resistance, increased hemoglobin A1. That is known as glycosylated hemoglobin. So interestingly, cirrhosis patients often have a higher abundance of oral cavity microbial strains such as Prevotella, Streptococcus in the gut microbiome, which are generally not present in healthy individuals. So you can expect that in a liver cirrhosis patients, the bacteria which is present in our oral cavity, that is the mouth buccal cavity, so that is also found in the patients having liver cirrhosis. So every bad bacteria can occur in your disease condition. It can increase the population of bad bacteria. So whatever be the metabolic disease, all the bacteria are interlinked to each other. It is not necessary that your bacteria is only present in your gut. It may also be present in your oral cavity. It may also be present in your different part of the body. So everything is linked to each other. Now one more thing. This is gut microbiota and NAFLD overall working mechanism. So, what are the gut derived factors and what are the liver derived factors? So, the gut derived factors are microbial component like LPS, viral bacteria DNA, because in our gut we are having bacterial DNA. So that bacterial DNA can act as a can act as a foreign which will initiate what? Inflammatory responses. Like we measure in our condition, in my experiment, I used to measure the eDNA. That is known as extracellular DNA, that whether I am having a eDNA composition in my overall extra polymeric substances. So if the concentration of eDNA increase, then you can say that the DNA is acting as a foreign material to initiate the inflammatory responses. Today only I just did this experiment. So whenever there is increase in the eDNA, that is the source is bacteria. Bacterial eDNA can also lead to inflammatory responses, because obviously it is not your genomic material, it is someone else genomic material. And if it is from the bad bacteria, then it will have some what it will have, it will have some genes which are responsible to produce toxins. Then the condition will be more fatal. So apart from that, microbial metabolites, if the microorganism is doing its normal metabolic machinery, like if it is a yeast, or if it is any bacteria like Klebsiella, or any bacteria that is capable of synthesizing ethanol through fermentation, so it will produce ethanol, it will produce acetaldehyde, it will produce TMA, and TMA is trimethylamine, SCFA, short chain fatty acid, and other secondary bile salts. Now, diet derived factors are ethanol, fructose, choline, and TMA. Diet derived factor means after consuming a specific food item or a specific nutrient, after its digestion, in which form it converts. So that is known as the diet derived factors. Host derived factors are primary bile, which our body produces by its own, salts, mucin, and what are the liver derived factors? So these are the primary bile salts, folate and deoxycholate, microbiota specific IgA, serum IgA. Other factors are bilirubin, phospholipid, cholesterol, fatty acid, lecithin. So all this bilirubin and biliverdin are measured in your liver profile test. Now, why this is important? So what are the environmental factor inducing the dysbiosis? So the major factors are, I have mentioned this so many times, diet derived factors. Diet derived factors are there, and apart from that, food xenobiotics. Like if you are, I have discussed this point in my previous classes, I remember that whenever you are consuming a processed food item, so all those processed food items are rich in preservatives, emulsifiers, and different coloring agents to make it look so shiny and glossy, colorful. So all are composed of different preservatives, different flavoring agent, and many more things. And apart from that, only sugar. So even if you are taking any packed tetra pack drink, like the common one is fruity, so in that fruity also you can see, after consumption of the whatever amount it is in the tetra pack, you can see there is a deposition of something like sticky mucilage kind of thing in the bottom of this tetra pack. So this is rich in microorganism, because it is a sugary drink. Obviously it has a large amount of sugar, so that sugar will initiate the growth of certain microorganisms, and that too it is in contact with different temperature. Like if you are keeping it at very high temperature, so it will be spoiled. So if you are thinking that this is a packed drink, you can drink it, so try to open it and see in its bottom, so you will not be able to drink it for the second time. So try to have fresh fruits intact, because juice will directly increase your sugar level. So if you are an individual having some diabetes kind of thing, if you are vulnerable to sugar, so or if you are pre-diabetic, you can have fruits, because if you are consuming fruits, you are taking the whole fiber, so it will not directly, it is not that its glycemic index is not that high. And if you consume the juice, so you are providing your body with only increased amount of glucose. So all these things are related to what we are consuming. So food xenobiotics is, xenobiotics means what? A food that is contaminated with something hazardous, something foreign material are there. So preservatives, emulsifier, additives, flavoring agents, and all these things are harmful for our body. Medication, if we are for a longer time on any particular medication, antibiotics, proton pump inhibitors, so water treatment and or contamination, chlorine. So when we are consuming water, so like in my area wherever I am living, so it is very high in arsenic. Arsenic is actually in Nadia, but wherever I am living, Kalyani, it is rich in iron. Like you can just see with your naked eye how much iron is involved in your tap and all those syntax tank. It is reddish in color, reddish or sometimes brownish in color. So I'm so worried about myself that what to do, but I don't have any other option. So we can have the uh, any good filtration system so that it can be eliminated. But this Kolkata, I have seen that the overall Kolkata or not just Kolkata, Kalyani and the nearby cities are heavily contaminated with the iron. I don't know from where it is coming. So you can have a very lethal consequences with this iron, because ultimately this is a metal, and if a metal is accumulated in our body in a very larger amount, more than the threshold limit of what our body can tolerate, so it will result in different fatal diseases. So many different type of cancers you can say, so many different metabolic syndrome you can expect. So water treatment, and because chlorine is there in our water, the municipality water is rich in chlorine because they chlorinate the water to make it safer for drinking. So if the chlorine concentration is increased beyond the tolerance level, anything beyond the tolerance level is always fatal, because there is some sort of threshold limits which is set for every living organism. For humans it will be different, for microorganisms it will be different, for plants it is different, for animals it is different. So all it depends on the our own genetic makeup. So if you are using anything beyond the tolerance level of your body, so it will harm you in every possible way. So the gut liver axis and the role of gut microbiome in the pathogenesis of NAFLD. Now you can expect different kind of inflammatory responses in your liver uh, if you are exposed to all these factor mentioned. Like if you are not taking a good diet, if you are always taking food xenobiotics, like food rich in preservatives, coloring agent, food flavoring agent, so you are taking only the xenobiotics. If you are taking medication for so long, if you are taking contaminated water, then you are inviting different kind of disease in your body. So you can see here all these things are these inflammatory molecules are directly into your bloodstream. So if they are present in your bloodstream, they can cause different kind of disease. So the gut microbiome altered by environmental factor induces hepatic steatosis. The dysfunctional liver fails to restore gut eubiosis, which result in a vicious self-perpetuating loop promoting NAFLD progression. So liver disease or any gastrointestinal disease, all it depends on our own foolishness. I can say this because if everything is dependent on what we are eating, so we, and then also we are not controlling our diet, we are consuming only junk, we are consuming only unhealthy food item just for Hello. Are you guys able to hear me?
Now we can.
Yes, ma'am. Now audible.
Okay. I don't know why these teams always create so much of mess. I just heard the sound that it got disconnected, because whenever I am doing the slide play, I am unable to see what is going on in that team profile. So I just heard the sound, that disconnect sound. So I just ended the slide play and I saw that it is already disconnected. So everything, every time it is only this thing. The best one is, I think, as per, I don't know about others, but I like Google Meet. It is very nice. Always the simple things are the best. Okay. So now a little bit more detail about this NAFLD. The different things which are going on metabolically, and what are the impacts of that thing. So the role, that is the increased intestinal permeability. So this condition we have already read that this is the leaky gut. We have already studied what is leaky gut. So leaky gut will do what? It will directly allow the inflammatic mediators, directly it will bath into your bloodstream. So if anything is in your bloodstream, we are having different kind of cells in our immune cells in our blood. So what they will do, they will consider them as a non-self, or you can say it will consider them as an antigen, and then it will start reacting towards it. So
Ma'am,
whenever
sorry to
there is an,
interrupt. Slides are
yes,
not visible, ma'am.
okay, wait,
Yes, ma'am.
okay, now it will be visible. So if anything is present in our blood directly, so our immune system is responsible for generating immune response towards it. So what it will do, it will consider them as non-self. So anything which is non-self to our body, our immune system will consider it as an antigen, that is antagonist or villain, and it will start reacting against it. So it will again increase the inflammatory mediators in our body. So whenever there is an increased intestinal permeability, that is known as leaky gut. So how this leaky gut condition appears? Whenever you are having dysbiosis and your cell tight junction is not working properly. So this is known as the leaky gut, and this will increase the level of inflammation. Inflammation will rise at its maximum. Choline metabolism. So gut bacteria converts choline to methylamines and therefore decrease in the choline, which will impair the VLDL formation, which is the reason of hepatic fat accumulation. Now, very low density VLDL is what? Very low density lipoprotein. So there are different kind of lipoproteins. In biochemistry, we can study HDL, LDL, VLDL. So this HDL is a good cholesterol, and this LDL, VLDL, all these are bad cholesterol. All it depends on the level of lipids it has. So VLDL formation, if it is impaired, then hepatic fat accumulation will increase. So whenever your gut bacteria is converting choline to methylamine, then there will be a decrease in choline. Choline is a protective molecule. So this is also going on in our body whenever we are having a dysbiosis condition in our body. And the most common one, we are studying this since first day, is CFA, short chain fatty acid imbalance. Excess acetate promotes the lipogenesis in the liver. Lipogenesis means synthesis of lipid. So alcohol production, some gut like I have taken this example, Klebsiella is one of the known genus which can synthesize alcohol. Like in our lab also we worked, I have also worked on this strain Klebsiella. So this is a very hyper ethanol producer strain. So it has capacity that it can produce ethanol from variety of substrate. So some gut microbiomes like Klebsiella produce endogenous ethanol, and that will increase the liver inflammation. Bile acid dysregulation. So whenever we are having imbalance in the bile acid produced in our body, so that will also affect the lipid metabolism and hepatic fat storage. So see, all it depends on the whether the lipid or the bad cholesterol is properly degraded or properly metabolized in our body or not. If they are not properly metabolized, they will be deposited. So if anything that is bad for a body is deposited for a longer time, so it will cause inflammation in your body. So this is the reason our liver is doing what? Our liver is accumulating these things slowly, and then the upper layer of the liver, or you can say the hepatocytes, are not able to do their normal metabolic functioning for what they are known to do. So these things are going on, like 10% liver damage, 20% liver damage, 30%, and over a long period of time, chronic liver disease means your more than 70% or 80% of the liver is damaged. So this is the condition that we can have non-alcoholic fatty liver disease also, and we can have liver cirrhosis also. But NAFLD is more common because it is diet related. It is related to dysbiosis, not only just alcohol related, but it is related to dysbiosis and diet. These are the two major contributors of NAFLD. Now we have to start the we are done with the NAFLD, obesity, and diabetes thing. Now, this is the third part of the third module. That is how our gut microbiome is related to neurological disorders. We have already studied this in second unit, like how our microbiota is related to different kind of psychiatric disease, if you remember. So these are some of the disease which you can expect. These are the neurological disorders, like our neurons are affected, our neurons communication and coordination is influenced if we are having an altered gut microbiome, that is dysbiosis. So you can expect autism spectrum disorder, you can expect Parkinson's disease, Alzheimer's, which is very common, sclerosis, anxiety and depression are the most common one. Even a student of class 10 has also a depression and anxiety wala thing. Epilepsy, stroke. So all these are neurological disorder which you can expect if you are having a altered gut microbiome. So if dysbiosis will increase, whose concentration will be increased? Clostridium concentration will be increased and bifidobacterium concentration will be decreased. So you can also have the GI symptom common in ASD. Like this is the major symptom of the ASD, autism spectrum disorder. Now, microbiota transplantation improve behavior in some cases. These are the therapeutic approaches. Like if there is an disease, obviously there will be a therapeutic options. And in Parkinson's disease, increased amount of Enterobacteriaceae and decreased amount of Prevotella. And in Alzheimer's has no such reported increase or decrease of any specific bacteria. So it will promote the neuroinflammation. And in multiple sclerosis, there will be decrease in SCFA producing bacteria. In depression and anxiety, lactobacillus bifidobacterium will be decreased. That is why we are recommended to have some probiotics rich in lactic acid bacteria. Like if you are having curd in your daily diet, so you will be having a good gut bacteria. Epilepsy, so apart from that, stroke. So all these diseases are the neurological disorders. Whenever you are facing any alteration in a gut microbiome, you can expect all these diseases if it is for a longer run. It is not acute disease. All these diseases are a very, very chronic stage. So this is not like if you are having an altered gut microbiome by eating something for 1 or 2 days, then you can expect Parkinson's. No, all these diseases are very chronic one. They are just a result of years of altered microbiome composition. So we will complete this. There are so many things we have to study, like the various kind of cancer. So we will study this from tomorrow. We have to finish this unit by the Friday, because two more units are left, and we have started from 24th was the introduction class. We have started from Monday, I think, Monday 26. So we have to finish it on time so that we can have 1 or 2 days MCQ session.